Hi everyone, this is the introduction to the digestive system. In this lecture, we're going to look at the major digestive processes. That includes motility, digestion, and absorption. We're also going to remind you of the macromolecules and how they're processed in the digestive system. So we're going to look at protein digestion and absorption, carbohydrate digestion and absorption, and lipid digestion and absorption. We will also introduce the anatomy of the digestive tract and the accessory organs of the digestive tract. So we're going to list and locate all of the digestive organs, look at the peritoneum and the layers of the abdominopelvic cavity wall, and also look at what it means to be retroperitoneal, as some of our abdominal organs are behind the peritoneum. We're also in the process of looking at absorption. We need to have an understanding of the microscopic layers, the blood supply, and the nerve supply of the digestive organs. So we'll look at those as well. Starting with an overview of the digestive system. The digestive system is responsible for the processing and uptake of ingested nutrients, the food that we take in, as well as fluids, vitamins, and minerals in the body. We're going to start with the oral cavity, move our way through the upper and lower GI tract, and then also talk about the accessory organs. We will introduce them in this lecture, and then we will have a separate set of lectures to go through the details of each of the processes of all of the GI tract and accessory organs. So, digestive functions. We want you to have a good understanding of motility, that's movement through the digestive tract, and how the smooth muscle coordinates that movement through the digestive tract. Also, mechanical breakdown and chemical enzymatic digestion in the tract. And then finally, absorption. How, once the molecules are broken down, how they get into the blood so that the body can use them. So first, motility. I love these Awkward Yeti comics. You guys know I'm an Awkward Yeti nerd. So here's the one about how coffee affects our digestive tract. We all know that, right? So motility in the GI tract involves involuntary waves of contraction and relaxation of smooth muscle in the digestive organ walls. If it's too fast, we can have malabsorption, or not enough time to absorb, and inadequate breakdown. That can lead to things moving too fast and diarrhea from the GI tract. If it's too slow, that can lead to the feeling of nausea and vomiting, and too much fluid then can be actually absorbed from the GI contents. And that can lead to constipation or inability to release the GI contents. The major types of movement we will see as we move through the digestive tract are peristalsis, which is the use of longitudinal and circular muscles to propel food forward through the tract. Segmentation, which is more of a churning and breaking apart using circular muscles. That's mostly done in the small intestine. And then mass movements, which are slow, coordinated contractions of large segments of smooth muscle. That's done in the large intestine for elimination of the digestive breakdown products through the defecation reflex, and we'll talk about those as well. Next is mechanical and chemical digestion. So when you hear the word digestion, I want you to think of breakdown. And then distinguishing between the two types of digestion is how that breakdown occurs. So mechanical digestion is mashing, grinding, compressing, physical breakdown. This is what we do when we chew, or when we have segmentation movements that are breaking things down into smaller pieces. The purpose of breaking things down into smaller pieces is to help them move through the digestive tract, but also to give it more surface area for the chemical digestion to take place. So chemical digestion is using enzymes. There's a lot of enzymes that are secreted by the digestive tract and by the accessory organs, mainly the pancreas of the digestive tract. Enzymes break down the macromolecules into their monomers or small parts. Proteins get broken down to amino acids. Sugars and starches, polysaccharides, get broken down into simple sugars, monosaccharides and disaccharides. Lipids or fats get broken down into monoglycerides or glycerol and fatty acids. Once the molecules are broken down into their small monomers, those small molecules can then be absorbed across the wall of the digestive tract. So small molecules, in addition to water, vitamins, and electrolytes, are all brought from the lumen of the digestive tract into the blood or the lymphatics very near the epithelial cells of the digestive tract. The primary location of absorption is the small intestine. But we're going to talk about the different types of absorption that can happen throughout the GI tract. The reason that the small intestine does the majority of absorption is because by that time, most of the digestive breakdown has happened. And the small intestine has a large surface area, the presence of villi and microvilli on their epithelial cells lining that lumen. All of that surface area provides a nice area for absorption of those contents from the lumen into the blood. There's membrane transport processes that are very important for this absorption to be able to happen. So just as it was important for us to remember our membrane transport processes in the kidney and looking at renal transport, it's also important to remember them in the digestive tract. So think about how molecules can move across these cells through the process of diffusion, through channels, through active transport through pumps and transporters, and vesicular transport using vesicles. Let's look at some examples. Here's an example of carbohydrate digestion. Some example of enzymes that break down carbohydrates. There's a salivary amylase, pancreatic amylase, and then brush border enzymes within the small intestine. They will slowly break down the carbohydrates into monosaccharides, and then those will be absorbed across the wall of the small intestine. So let's zoom in on this diagram so you all can see it a little bit better. So here we start in the lumen of the small intestine, where, for example, we have amylase breaking down a starch into small sugars or disaccharides. And then those disaccharides will be broken down by the brush border enzymes into monosaccharides. Those monosaccharides, for example, glucose, can then be brought in through transporters, for example, the GLUT transporters that are present on the epithelium. They will then be transported inside the epithelial cells of the small intestine, travel across the small intestine, and then to the other side, the basolateral membrane, where they will also then be carried across the membrane and then can be added to the blood. So absorption of carbohydrates starts with the digestion and ends with the movement of those digested materials across the cells and into the blood. Protein digestion is really similar. Here's some examples of protein breakdown enzymes: pepsin, trypsin, chymotrypsin, carboxypeptidase, and also some brush border enzymes found in the small intestine. So let's look closely at protein digestion examples. So we'll zoom in here. So here again, we're in the lumen of the small intestine. We have these proteases, trypsin, chymotrypsin, etc., that are coming from the pancreas. They're going to break down the protein into amino acids. or small peptides. And then those small peptides can be broken down even further by the brush border enzymes. Those brush border enzymes will break them down into small peptides or amino acids, and then those can be absorbed into the epithelial cells through amino acid transporters. Once they're in the epithelial cells, then they will travel across to the basolateral membrane and then be carried out of the cell and then transported into the blood. So carbohydrates and proteins, notice, have similar processes for digestion. What you need to know really is the name of the enzymes that are slightly different and the location of where those enzymes are coming from. We'll learn that as we go through different areas of the GI tract. Lipid digestion, on the other hand, is a little bit different. Lipid digestion first requires emulsification of using bile salts from the liver. So that's because the lipids are very large and they also float away from the epithelial cells. So we need to break them down into their smaller parts to open up more surface area for their digestive enzymes. That's done mostly through bile salts from the liver. So lipid digestion starts with emulsification. And then we have the pancreatic lipase that can break it down in the small intestine. Finally, once they're broken down, they will be packaged into vesicle-like structures called micelles. And those micelles are what's responsible for transporting those packets of lipid across the epithelial cells. They get repackaged into another group of vesicles called chylomicrons. And then instead of entering directly into the bloodstream, most of those packaged lipids are actually going to enter into the lymphatics through the lacteals. I've tried really hard over the years to figure out why can't they just go straight into the bloodstream. If you guys have any research on this, let me know. As far as I can tell, it's a size issue. They're simply too big. So they're transported into the lacteals first, then brought to the bloodstream through the hepatic portal system to the liver to be processed. So lipid digestion is unique to carbohydrate and protein digestion. But these are the three main patterns of digestion and absorption I'd like you to be familiar with. Now let's look a little bit at the anatomy of the digestive tract. One thing to note also is that we have visible body set up this week so that you all have a lab number 4, digestive system lab. I really recommend, because of the importance of the digestive system and because there are so many different organs to get straight, that in addition to what I tell you in the next few minutes, you also go through your virtual lab number 4 and look at the anatomy of all of these digestive tract organs. So the digestive organs of the GI tract are directly connected to each other and have hollow lumens for the passage of food material. So essentially, they're open to the environment, meaning that when we open our mouth to bring food in, we're bringing that food and fluid in from the environment. And then the digestive tract is open throughout as the food and nutrients passes through it. We start at the oral cavity, move down the throat or pharynx, into the esophagus, and then down into the stomach, where it can be stored for a short period of time, and then into the small intestine, where we have the majority of digestion and absorption taking place. Final processing in the large intestine before the final elimination of the food waste, which is defecation. We also want you to appreciate the accessory digestive organs and their very important role in the digestive tract. Don't think that accessory means less important. The only reason we call them accessory is because they're not part of that hollow tube system of the alimentary canal or the GI tract. Instead, they're separate from the GI tract and secrete into the GI tract, starting with the salivary glands secreting saliva into the mouth, and then the pancreas and the liver secreting enzymes, digestive juices, and bile salts, respectively, into the small intestine. The gallbladder also helps with bile secretion. So the liver makes the bile, the gallbladder stores it, and can release it through the bile ducts also, along with the liver secretion of bile into the small intestine. When we look at the organs of the GI tract, just like we looked at the other organs in our body systems, we see that there's a serous membrane that surrounds most of them. This is the peritoneum. There's a parietal peritoneum that lines the wall of the abdominal cavity, and a visceral peritoneum that is right on top of the organs. The peritoneal cavity then contains a serous fluid like many of our other serous membranes throughout the body. What I want you to notice though, and I wish we had time to go through more of the embryology, but what I want you to notice is that those are mostly going to be in the anterior and middle portions of the abdominopelvic cavity. The more posteriorly, or in the posterior abdominal wall, are actually going to be behind the peritoneum, and those are called retroperitoneal organs. So more specifically, the peritoneum is divided up into specialized folds that are together around organs to help to suspend them and hold them in place. These are called the mesenteries. It holds those digestive organs to the body wall, contains lots of blood vessels, lymphatics, and nerves, and also stores fat. The organs that are not within the peritoneum are behind it or below it. The ones behind the peritoneum are called retroperitoneal. And probably a good idea to just go ahead and memorize the list of organs or parts of organs that are retroperitoneal. So that includes the pancreas, the early part of the duodenum, the rectum, and then as we move further down, we have subperitoneal organs, the bladder, in females the uterus, and also more retroperitoneal behind the kidneys. Here's a list of specific mesenteries that may come up later in some of your anatomy studies. We have the falciform ligament, which attaches the liver to the body wall and separates the right and left portions of the liver. We have the lesser omentum, which attaches the upper curvature of the stomach to the liver. We have the greater omentum, which is actually this huge apron-like structure that attaches to the bottom of the stomach and actually covers over all of the abdominal organs. The greater omentum is also referred to as the fatty apron because of the way in which it sort of hangs down and moves around on top of the abdominal organs. Then we have the mesentery proper. This is dorsal and part of the small intestine, attaching the small intestine to the back body wall and also found linking the portions of small intestine together. Then we have the mesocolon, which attaches the transverse and sigmoid colons to the back body wall or the posterior abdominal wall. There's a lot of blood supply to and from the abdominal organs. Think about it. This is the place where we're having the majority of nutrient absorption. This circulation of blood supply to the digestive organs is going to allow the nutrient absorption to happen, but also supply those organs with the necessary nutrients that they need to be active as they are through all of these processes. Splanchnic is another word for organs. So the splanchnic circulation is the circulation to all of these abdominal organs. And they're branches of arteries from the abdominal aorta to the digestive organs. Then we have this really nutrient-filled blood that's going to be processed in the liver. That nutrient-filled blood is going to go to the liver first before the general circulation. And that circulation to the liver from the digestive organs is called the hepatic portal circulation. This is blood draining from the abdominal organs to the liver before it gets to the inferior vena cava. And we'll talk about this more in our liver lecture. There's also a huge nerve supply in the digestive tract. This is called the enteric nervous system. There are so many nerves within the digestive tract that some even refer to this as the gut brain. And it's a semi-autonomous network of neurons that have pacemaker cells running the motility of the digestive tract, setting the pace for the peristalsis and the segmentation and the mass movements and local reflex arcs within the digestive tract. So this nerve supply is incredibly important for the motility of the smooth muscle within the digestive tract. Similar to the heart, it also gets input from the autonomic nervous system, which can modulate its motility. Remember that parasympathetic is the rest and digest portion of the autonomic nervous system. So it is the supply from the vagus nerve and the pelvic splanchnic nerves that are going to be the parasympathetic input, increasing digestive tract motility and activity. So I'll say that one more time. Parasympathetic input increases digestive tract motility and activity. Sympathetic input through the thoracic and sacral splanchnic nerves are going to be inhibitory. They're going to slow down and block digestive tract function. As we think about these functions, I also want to show you the general digestive histology. I know you've done much of this in your prerequisite courses, so we're not going to harp on it too much, but I want you to have an understanding of the four main layers within each digestive organ wall. That is from the innermost lumen to the outermost layer. So if we zoom in and we look at the center of one of these hollow organs, this is kind of a general view of what we would see, say, in the small intestine. We have the lumen where the food is going to move through. And then right up next to that lumen is the mucosa. The mucosa is the epithelial layer of the digestive tract. It's called mucosa because many of these areas secrete mucus. But I also want you to hear secretion and think not just mucus, but also enzymes, fluids, water, salt, many secretions that can be possible from a mucosa. In the stomach, stomach acid, so many different secretions possible from the mucosa. And then there's a layer deep to the mucosa, which is the submucosa. That submucosa is an area that is just filled with glands and blood vessels and specialized structure that support functions in particular regions of the digestive tract. Remember, for all of this secretion to happen, we need lots of blood supply, nerves, and glands, and many of those are in the submucosa. Then we have some muscle layers. There's a muscularis mucosa that is a small layer of smooth muscle between the mucosa and the submucosa. But it's deep to the submucosa, moving more towards the outside, that we have those big layers of smooth muscle, usually circular and longitudinal layers, and they're going to be responsible for the motility through the digestive tract. So that's called the muscularis externa, large layers of smooth muscle helping with motility here. And then finally, depending on where we are, there's going to be an outer layer. If it's part of the peritoneum, that outer layer is the serosa we just talked about. That's the visceral peritoneum. If it's not part of the peritoneum, if it's a retroperitoneal organ, then it's going to have an adventitia, just another layer of connective tissue. So please look at these four, four. Please look at these four layers: mucosa, submucosa, muscularis externa, and serosa layers. Those are really key for the function of all of the digestive tract organs. And we'll talk about the functions for each organ and how their layers may be different in the following lectures. Here's one last view, taking a little slice from the digestive tract organ to show you those four layers in cross section. This one is a little less busy than the previous slide, so let's look at them again from here. We have the mucosa, which is all of this epithelial tissue, the submucosa containing the blood supply glands and nerves, and the muscularis externa containing that smooth muscle, and then finally the last connective tissue layer, the serosa or adventitia. All right, that's it for the introduction. Let me know if you have any questions, and we're going to move on now to each of the different organs of the GI tract and the accessory organs. See you in a minute.